Hi everyone, my name is Brian Sanchez. I'm a student at UMass Medical School, and today I'm going to be talking to you guys about the different kinds of brain research. What I'm hoping I can teach you guys today is kind of why scientists use various models to study the brain and how each model will kind of tell us something different and makes each of them unique in their own way. So let's get started. So the first model that we're going to be talking about is obviously animal research. So the reason why scientists use animals to study the brain is that they are biochemically and genetically similar to humans. So what I mean by that is that there are certain animals that contain the same chemical molecules, metabolic pathways, and genes that a person has that can be easily studied to learn more about how the human brain functions. And more specifically, if you look on the slide, we have mice, which actually share many neurotransmitters with humans. Cats and rabbits actually are being used for models of vision. Flies have similar features in their nervous system, and certain fish that lay transparent eggs can teach us more about how developmental processes happen. Now I'm going to talk a little bit about how these animals can actually teach us about chemical connections in the nervous system, and more specifically with Alzheimer's disease, which is the death of neurons containing the neurotransmitter acetylcholine. And I'm going to talk about this a little bit in the next slide, but we're going to talk about how these new staining techniques can teach us ways to look at pathways and connections between brain areas, and along with that, identify neurons containing and using certain neurotransmitters. The two types of staining techniques I'm going to teach you guys a little bit about are Nissl stains and Golgi stains. So in the Nissl stain, what's happening is that you are staining specifically for nucleic acids, which are either RNA and DNA, and these are it only stains specifically for RNA that is expressed outside the nucleus. And what you can see is that it specifically stains only for cell bodies. So if you look at the slide, you can see that where all the kind of dark blue and purple areas are the cell bodies that are stained specifically staining. The second type of staining is Golgi stains, and these stains are a little bit better because in these you can see full neurons, and that in the Golgi stain you're staining the cell body, the dendrites, and the axons. I want to talk to you guys a little bit about how these animal models not only can teach us more about the pathogenesis behind disease, but also how it can lead to new treatments for diseases. I want to talk about Parkinson's disease and how it has been specifically studied in rabbits, mice, and pigeons. So while they were studying Parkinson's disease in rabbits and mice, they were able to find that the neurotransmitter dopamine was being depleted in these mice that had Parkinson's disease like symptoms. And in pigeons, they actually found that dopamine is highly concentrated in the basal ganglia, which gave us a place of, okay, that in these animals that display Parkinson's like traits, that the dopamine is being depleted, and an area in which dopamine is highly concentrated in the brain is in the basal ganglia, which was found not through human studies, but actually through rabbits, mice, and pigeons. After learning these facts, scientists decided that we should try treating Parkinson's disease by making more dopamine in the brains of humans, and that's currently what's being used as a treatment now is something called levodopa, which is the treatment for Parkinson's disease, and its mechanism of action is that it converts dopamine in neurons. Another disease that scientists are trying to learn more about by using animal models is drug addiction. And specifically, scientists are interested in learning about rats and how rats can become addicted to the same drugs as humans. So, what we want to learn more about are the parts of the brain that are leading to this addiction towards drugs. And what scientists have found is that it's mostly in two areas, and that's the ventral tegmental area, which is where dopamine neurons can release and lead to the reward pathway in these mice that stimulate their happiness and will cause addiction. Another thing that we learned is how the ventral tegmental area actually communicates with the nucleus accumbens to enhance the reward pathway in these mice. So just by studying these mice, we actually learned a lot about the reward pathway and how it's activated by natural rewards like food, water, and sex. And along with that, people are trying to look into how we can treat people with drug addiction by using drugs to mimic or block neurotransmitter function. Along with diseases, these animal models can actually teach us a lot about just the regular function of the brain, more specifically learning and memory. And something that I didn't expect when I first learned is that sea slugs actually taught us about short-term and long-term memory. So they taught us and how the synapses are involved in both short and long-term memory. So we figured out more about short-term memory by studying how sea slugs actually how their reflexes work when you induce them with a certain stimuli, and after you induce them with a stimuli, it's retained as short-term memory because of stronger synapses. Long-term memory was learned through um, the activation of genes, in that when you hit them with these very strong stimuli, that they would activate certain genes, which would increase or decrease certain protein level expressions. So what that taught us is that we're getting growth of new synapses in these sea slugs that are leading to long-term memory. Along with learning and memory, these animal models have also taught us a lot about the critical periods of development. So, critical periods are a period of time during which experiences guide the development of a system. And by a system, we mean, animals, either cats, monkeys, or rats. And how these circuits, after they are developed, can no longer be easily modified, that these are very fixed behaviors within different species. In monkeys and cats, we have learned about critical periods for modifying visual circuits, while in mice, we have learned about what changes in the brain are required to prevent rewiring of the brain. I want to talk about how experiments with monkeys and cats help determine a treatment for amblyopia. So, amblyopia is a condition in which the vision of one eye is greatly reduced because the eyes just aren't working that well together. And it's been discovered in these monkeys and cats that it starts early in life before the age of 8, and how studies in mice they have been able to remove factors preventing rewiring and cure older amblyopic mice. So hopefully we're hoping that one day, we'll be able to do the same things in humans. So now we're going to step a little bit away from the animal models and just kind of talk a little bit more about how the brain can be studied through its biochemical pathways and things like that. I'm going to be talking to you guys about sample research methods and how neuroscience can be studied through all different types of different disciplines. And more specifically, we're talking about microdialysis, which is how to measure particular amounts of brain chemicals in a specific brain area. Doing this is very informative to scientists because it can help visualize brain activity and track nerve connections. And the way we can do this is by injecting radioactive amino acids into brain cells in tissue culture. So you just have your brain cells that you can grow up, inject them with these radioactive amino acids, and look for activities in nervous system to show up on film. And along with that, we can also inject horseradish peroxide into these brain cells that can be taken up by nerve fibers and can then be identified under a microscope. I'm going to talk to you guys about how electrophysiology can be used to learn more about the brain and how it functions. Electrophysiology is the study of the electrical properties of neurons. More specifically, we want to learn about action potentials, and action potentials are the way in which neurons communicate. So if you look at the graph to our right, you can see that these neurons have membrane potentials, and what happens is when a neuron fires off its electrical properties, that you get depolarization followed by repolarization, and then you get a small dip of hyperpolarization where the neuron can no longer fire and it has to go through a refractory period. These action potentials will eventually lead to something called long-term potentiation, which is the cellular event that makes memory and learning possible. So in this slide, we're going to learn about how electrophysiology is directly studied in patients. And so you can see that electrophysiology, you can record auditory brainstem responses to sound, and you can even diagnose hearing loss in infants through this. And that what they do is that in a patient, you have various electrodes that you can place on specific parts of the head to record neuron responses to sound, and then analyze your time lapse between when you administered the sound versus when the patient's action potentials were fired. Now we're going to step away from electrophysiology and move on to imaging. There are 7 main types of imaging machines that we use, and I will go over each of them individually. The first type of imaging that we're going to go over that doctors use to learn more about patients is computed tomography or CT scans. And the way that CT scans work is that it combines multiple X-ray images to form a 3D image of our brain. And the tomography is a method for producing a 3D image of an internal structure by the observation and recording of the differences in the passages of waves and energies on those structures. So in neurology, these are primarily used to diagnose head trauma and bony lesions within the brain. The second type of imaging that is used to learn more about patient brains is positron emission tomography or PET scans. And the way that these work is that it measures metabolism, blood flow, and oxygenation of the brain. In that it uses radioactive substances, and that specifically is a positively charged particle, which is called a positron, that emits radiation. So the way it works is that you have these radioisotopes which you can inject into a patient's bloodstream, and it will circulate to the brain, the radioactive isotopes, which will accumulate in the hard working areas of the brain proportionally. And from there, you can then use a computer image to show which levels, which areas of the brain are being used for specific exercises. So what are the uses of these PET scans? And you can really see the best things to use them for or is if you're looking for increased metabolism in the brain, either through cancer or a seizure, or decreased metabolism of the brain, either through brain death or memory loss. And this research is all being done right now with PET scans and looking to see how mind-altering drugs or different sensory or social experiences can lead to various metabolism differences within the brain. Along with that, we can also look to see if there's a difference in neurotransmitter releases in different patient brains through PET scans. The next type of imaging I'll be talking about is magnetic resonance imaging, or MRI, which are probably the most famous type of imaging. And the way that these MRI scans work is that they use magnetic fields to excite and detect hydrogen atoms. And as you know, I hope most of us know, is that when you have is that there are many hydrogen atoms in water and fat. These MRI machines can then create 3D images by using the hydrogen distribution within organs and structures of our body. And what we're kind of more specifically focused on is looking at, how these hydrogen distributions occurs in the brain. On the bottom of the slide, we have some images from an MRI, and what you can see in these images that the levels of brightness within the brain varies, and these brightness actually correlate with less hydrogen. So in tissue that has a lot of water and fat accumulation, you get a darker image, while tissue which contains little to no water, like bone, you get a brighter image. So these images are actually very informative to us, and they can tell us more about when structural abnormalities will first appear over the course of a disease, or how they can affect subsequent development, or even how their progression correlates with things like mental and emotional aspects of disease. Diffusion tensor imaging is a different type of MRI procedure in which you can use diffusion rates of water, which tend to be higher along the fiber tracts of our brain, and assess the path of fiber tracts in the brain. MRI imaging is very helpful with helping doctors determine what type of disorders are in a patient. More specifically, you can look at white and gray matter specific disorders like infection or dementia by using MRIs. Along with these infections and dementia, you can also look for more subtle disorders like brain tumors or seizures by using an MRI. Along with the clinical practices of MRI, you can also do research with MRI, like how white or gray matter develop over time and the early and late stage characteristics of a disease. I'm going to talk a little bit about the procedure that a patient will go through when they get an MRI. So it first starts with a patient lying inside a hollow cylindrical magnet, and they are exposed to a powerful magnetic field, which then, so the background of the magnetic field will line up all atoms of the brain, and then you get a second magnetic field at a different orientation turned on and off many times per second. At certain pulse rates, certain atoms resonate to line up with the second field, and when this second field is turned off, atoms swing back in their original position, creating a signal. This signal can then be converted into images. The next type of research I want to talk about that's being used to study the brain is magnetic resonance spectroscopy. So in MRS, it's the same type of machine as an MRI, but now we're not looking at concentration of hydrogen atoms, but instead we're looking for concentration of specific chemicals like neurotransmitters in different parts of the brain. And using MRS has already contributed to a knowledge of different things like brain development and aging, Alzheimer's disease, schizophrenia, autism, and how strokes develop. Along with the MRI, we also have the functional MRI or fMRI, and these are one of the most popular neuroimaging techniques today, and they're really popular because you can compare the brain activity during rest and active conditions. And the way it works is that it combines anatomical imaging of the MRI with detection of increases in blood level oxygen. By using these detections of increases of blood level oxygen, you can actually look at the neuronal activation that's associated with these increased blood flows, and it can be used to study a wide range of brain functions. And along with studying the brain, there's a lot of clinical use of these fMRIs, that you can determine the riskiness of a brain surgery. So by looking to seeing what's a more active part of the brain versus what's not, you can determine how risky that surgery will be. Along with that, you can identify functional side effects of other disorders. The way magnetoencephalography or MEGs work is that they reveal weak magnetic fields created by neuronal activity, and these are really useful because they can help us track rapidly changing patterns of neural activity. Along with it, you can also track how long neuronal activation is sustained. And what's really cool is that you can kind of have this power duo between an fMRI and an MEG, and you can look at areas of the brain activity while an fMRI does a task, and see which part of the brain then becomes active through an MEG. In the clinic, MEGs are used mostly for electrical disorders like epilepsy. If you look at the bottom right hand of the slide, you can kind of see how these MEG works, and that in the left, so it's graphed over time, and if you look at the most left one, so you can see the neuronal activity just prior to doing something, and as you start doing it, you can see that area start to light up, meaning that this area of the brain is that the neurons are being activated in that area of the brain during a certain activity. Now I'm going to change from talking about electrical imaging to more optical imaging and other techniques. And the way optical imaging works is by shining weak lasers through the skull to visualize brain activity. And the good things about it is that it's really inexpensive, it's portable, so you can do just about anywhere if you bring the equipment, and it's safe, and it's also safe to do. Near infrared spectrometry, or NIRS, works by shining lasers through the skull at a near infrared frequency, and pretty much what happens is that when you shine these lasers, the blood with oxygen in your brain will absorb the lasers and refract it back at a different frequency than what was absorbed. Along with NIRS, there is also event-related optical signals, And this works by recording how light scatters in response to rapid cellular changes when neurons fire. Transcranial magnetic stimulation, or TMS, works by inducing electrical impulses in the brain, which will then disrupt activity in that certain area in the brain and give you a virtual lesion. These virtual lesions actually can help us a lot in the clinic, in that by using these TMS, we can diagnose circuit disorders such as stroke, multiple sclerosis, ALS, and movement disorders. Earlier, I talked about how we use animal models because they share very similar genes with a person. And I want to go a little bit more into what genes are. And genes are these short sections or sequences of deoxyribonucleic acids, or DNA. And there are four main types of bases that DNA can have, either adenine, cytosine, guanine, and thymine. And the way that this works is that um, adenine will specifically form hydrogen bonds with thymine and form an AT pair in your double helix. DNA strands are visualized as double helix structures within the 23 chromosome pairs in every human cell. And humans have about 20,000 to 25,000 pair of genes. More than 7,000 disorders are suspected to have a genetic basis, and by that I mean that there are these mutations that occur, which are changes in DNA such as misspellings or the correct amount of DNA within a person. These mutations can then prevent a gene from functioning properly or contribute to disease. There are two main ways in which we kind of study genes, and that's either through a genetic linkage study or through a chromosome microarray. In a genetic linkage study, what happens is we study families and large groups of unrelated people with certain conditions, and we try looking for positions in chromosomes and locating genes that lead to these various diseases. In the chromosome microarray, instead of looking at these big populations, we're now just looking at one individual. And we want to look carefully at the overall chromosome makeup of that individual and determine if there are segments of chromosomes, sometimes containing multiple genes, that are either missing or are more present than usual. So they're either deleted or duplicated. These help us identify rearrangements of chromosomes associated with disabilities and disorders. So why do we go through all this effort to learn more about genes? And that's because knowing genetic information can help clarify diagnosis, help understand the cause of symptoms, and even understand the chances of a condition affecting or being carried by a family member. I want to talk more about specific genes that are associated with neurological conditions and disabilities. So the first one I want to talk about is the HTT gene, or Huntington's gene. And in this gene, if you have a single point mutation, it will lead to Huntington's disease in a patient. Next is the RB1 gene, or the inherited retinoblastoma gene. And this mutation in this gene is very rare, but it is it causes a highly malignant childhood eye tumor that can lead to blindness and death. The DMD gene, or the Duchenne muscular dystrophy gene, is an X-linked gene, and what happens when you have a mutation in this gene is you get progressive muscle disease. The FRM1 gene will lead to Fragile X syndrome, which is a very common cause of inherited intellectual disability within males. Rarely a child might have a genetic mutation that did not come from either parent, but it's more likely than not to have come from one parent or the other. The LIS1 gene is really important because it helps tell the brain how to grow, and mutations in this gene will lead to smoother brains than normal and sometimes even seizures. I'm going to briefly talk about how some of the genes that are associated with conditions that include autism or autism-like syndromes. Mutations in the genes TSC1 and TSC2 will lead to something called tuberous sclerosis complex, and this is highly associated with autism-like symptoms. Another gene mutation that is linked to autism-like symptoms is MCP2, which will cause Rett syndrome. And deletion of certain parts of chromosome 16 can lead to neurological conditions, including autism in some individuals. But as of now, no gene or set of genes can be attributed to the majority of autism cases. Now, scientists are determined to sequence people and learn more about the genes and the DNA that make up the human body, and we have been determined to determine the functional sequence of all 20,000 plus genes in a person, as well as the remaining DNA thought to regulate genes. We want to be able to explore which variants are associated with a disease or disorder. And we're doing this through something called next generation DNA sequencing. And through this next generation DNA sequencing, we've been able to identify a gene called MLL2, which is responsible for Kabuki syndrome. And Kabuki syndrome causes this inherent intellectual disabilities and certain abnormal facial features.